Well, thanks everyone for joining us today, and we are excited about everyone's participation. And we want to encourage you to submit your questions throughout the webinar today as you can think of them. As Amy indicated, our panelist, Andrew, actually used to work as a fellow and research microbiologist for the CDC before joining us at ARL and now as the supervisor. Tiffany has been with us for many years and frequently cited and published and heads up our senior microbiology and does a lot of the technical and scientific projects. And then James has a tremendous amount of experience in the microbiology. I declare a conflict of interest, real or apparent, and financial interest in any company, product, or service mentioned in this program, including grants, employment, gifts, stocks, holdings, and honoraria. Today we're actually going to cover a review of some of the USP microbiology general chapters. In addition to the traditional microbiology test, we'll be covering some of the biological tests such as endotoxins and assays, as well as some of the informational chapters that can be applied and frequently referred to in the USP. We'll also be discussing the USP non-sterile drug product testing requirements, as well as the sterile drug product testing requirements. In addition to the traditional information, we are adding areas of environmental monitoring, and then the investigation and microbial identification will be discussed and covered, as well as there's some good literature references provided for your use at the later date. The agenda: at the conclusion of this program, the participant, pharmacist, or technician should be able to explain the testing requirements based upon the USP microbiology test general chapters. Number two, they should be able to identify the microbiological tests used in managing the production of non-sterile drug products, interpret the sterility test results and assess sterility test limitations, and identify areas of pharmacy operations that require environmental monitoring. First, let's explain some of the testing requirements based upon the USP microbiology test general chapters. To set the basis, we know that the scientific USP is a scientific nonprofit organization that sets standards for quality, purity, identity, and strength. Those terminologies have been discussed and covered quite a bit in the past. The USP's drug standards are enforceable by the FDA, and the U.S. Federal Food and Drug and Cosmetic Act designates the USP as the official compendia for drugs marketed in the United States. In general, we know that the monographs consist of a series of tests, procedures for those tests, and then as well as acceptance criteria. We will cover some of the acceptance criteria in several of the chapters and tables which you'll see are presented today. We know that the general chapters less than 1,000 are enforceable, such as 71, 51, 61, 62, 797, and those general chapters that are greater than 1,000 are for informational purposes and not necessarily enforceable. To start our review of the USP microbiology general chapters, I've provided the tables here that list the most common tests that are used to verify the microbiological quality of sterile and non-sterile drug products. Some of these tests are applicable to in-process product testing as well as finished product testing, and I'll be differentiating those details throughout the presentation. And although the bacterial endotoxin test is not technically a microbiological test, it is included here because it generally falls within the discussion of microbiological quality due to the fact that endotoxins are a byproduct of gram-negative bacteria. These chapters are intended to give information to support compounding practices as well as quality testing. Along with a quick summary of these chapters now, I'll be referencing the chapters throughout the presentation where they're applicable. Also, these chapters are not enforceable. However, there is useful information with them, and I recommend becoming as familiar with these informational chapters that are, as well as those that are enforceable, or those that are numbered less than 1,000. The first one in the list is 1111. This chapter provides us with some guidance on the acceptance criteria for non-sterile pharmaceuticals based on the route of administration in which it's given to the patient. It also includes suggested acceptance criteria for products intended for the pharmaceutical use, and this can be referenced for raw products, excipients, or in process bioburden testing. The next chapter, 1113, discusses microbial identification and when different levels of identification are appropriate, which we'll discuss toward the end of the presentation or the webinar today. USP 1115 gives us information on bioburden control during non-sterile drug manufacturing. And as I'm using the term manufacturing, one can imply the compounding. We know there's a regulatory and there's a differences between compounding and manufacturing. But in general, when we're talking of manufacturing, these are guidance, these are chapters that are beneficial, whether you're compounding or you're manufacturing, just basic good practices in microbiology testing. It's a very nice diagram demonstrating possible sources of introduction of bioburden in 1115. And would be useful for validating the manufacturing processes as well as investigations relating to the quality of the product. The next one, 1116, is the chapter that gives us the most information about environmental monitoring for the aseptic processes and sterile compounding. So 1116, with the emphasis on environmental monitoring, it's a really good chapter to look at. 1117, it's also a good chapter focusing on microbiology specifically. It's applicable to some aspects to every subject that we'll discuss today. It provides details for quality control testing of microbiological growth media, including but not limited to growth promotion testing. It also discusses maintenance of challenge microorganisms and how to properly set up a microbiology lab to be used for pharmaceutical drug product testing. I'd recommend that everyone read it and become familiar with Chapter 1117, as this chapter provides information that serves as a good foundation of all USP microbiology testing requirements. 1211 chapter covers the sterilization and the sterility assurance of the compendial articles. 1227 is a chapter that's referring to the validation of microbial test methods. It focuses on microbial recovery from pharmaceutical products, as this is the main objective during any microbiology test validation. Chapter 1229.3 is the monitoring of the bioburden. In this chapter, it discusses the importance of process bioburden testing, monitoring, and sampling, and identification of microorganisms that are recovered, as well as some of the more on bioburden control. In contrast, whereas 1115, that chapter actually focuses on the process bioburden of products that will be sterilized by methods such as filtration, steam sterilization, and radiation. And let's get into more of the detail. In USP 51, the blue section of each chapter is a basic statement of the purpose or intent of that particular chapter. You'll see in 51 it demonstrates preservative effectiveness for all injections such as topical, oral products, and antacids. So I'm thinking of antimicrobial effectiveness as a method of measuring the effectiveness of the antimicrobial in the drug product or preparation. We'll review each microbiology test chapter in numerical order, starting with this USP 51. USP 51 is used to verify the effectiveness of the antimicrobials or preservatives that have been added to that drug product to aid in maintaining its microbiological quality during the patient's use. As such, the microorganisms used to challenge the effectiveness of the antimicrobials are selected based upon the likeliness of the specific genus or species that may contaminate that product while it is being used. The antimicrobials and preservatives are used in both sterile and non-sterile products. Therefore, this text chapter is applicable to both. An example of the product that must be tested for antimicrobial effectiveness is a multi-dose vial containing a sterile drug product. Another example I like to use And this one would be applicable to non-sterile drug product as a scan or a face cream. Sometimes these are packaged in small tubes that require the end user to use their hands to withdraw the product from that container. And the purpose of the preservatives in this type of product is to ensure that microorganisms from the user's hands cannot grow once they have been reintroduced into the product or introduced into the product. This test is used for finished product testing. This chapter 51 specifies that the procedure and acceptance criteria for effectiveness applies to a product in the original sealed container in which it was distributed by the manufacturer. This means that the test must be performed on the product that is packaged in its final container. The label on the final container must also declare the antimicrobial preservative that's been added to the product itself. Method suitability must be performed for that specific formulation to ensure recovery of a microorganism during the testing. In addition to the information provided in 51, one would reference USP 1227, which is that validation of microbial recovery from Pharmacopeia articles for requirements to verify that the method is capable of detecting microorganisms in the presence of the product. Growth promotion testing must also be demonstrated for the media that will be used for this testing, and you'll see these common threads in each of these chapters that we're discussing today. To perform this test, USP 51, the product is divided into separate aliquots to be tested by the selected challenge microorganism. USP 51 provides a list of microorganisms to be used. However, this list is not exhaustive. The drug product manufacturer should also evaluate the necessity of other challenge microorganisms based upon three things: the nature of the individual excipients, the manufacturing process, and the details of the use of the finished product. For an example, an excipient of the product may be known to have growth promotion properties for a particular class or genus or microorganism, and you'd want to ensure that that antimicrobial are effective against this class or genus. Another example might be that the product is designed for a specific route of administration or a specific type of patient, where a microorganism that could be potentially introduced into the drug product might be considered objectionable. This is Table 1 in USP 51, and these are showing the categories and the description. So Category 1 is the injections and other parenterals. Category 2 is discussing the topicals. 3 is other oral products such as antacids. And then other products other than antacids. And then Category 4 is antacids. This is an article that has been included that was authored by Dr. Vu and then some students. But it's a good article on antimicrobial effectiveness testing, and I believe Amy has provided those for a resource. And then at the back of each of those articles are a lot of references within the article that goes back to the source documents. USP 55, it provides confirmation of the spore count on or in a biological indicator. Basically, the biological indicators are spores containing strips or ampules that are used during that development or validation process. These viable spore count test is performed by suggesting that microorganisms contained in the biological indicator conditions that encourage their growth, followed by subsequent incubating and counting the recovered colonies. USP 55 gives us the testing information that is required for biological indicators that are used during the sterilization process, in addition to other tools such as data loggers or temperature probes. Biological indicators are used routinely as a verification of adequate sterilization processes. USP 55, as an example of this, the bacteria Geobacillus stereothermophilus is used during verification of a steam sterilization, or Bacillus subtilis is commonly used for verification of dry heat sterilization. The population of the spores within the biological indicators would also be consistent with the level of sterility assurance that is desired for the sterilization process. Spore-forming bacteria are used for this application process because they are the most resistant form of bacteria to a sterilization process due to their ability to protect themselves by forming a spore that aids in their survival to unfavorable conditions. Let's look at some microbiological tests that are used in managing the production of non-sterile products. Non-sterile drug products manufacturing should include in-process bioburden testing. It is important to determine that the in-process bioburden does not have the potential to have an adverse effect on the finished product. This includes the product attributes, such as excipients and their potential to promote growth of microbial or microorganism populations, as well as the route of administration of the product and the demographics of the target patient population. In addition, the population must not have the ability to cause quality-related issues in that product, such as the ability to decrease the treatment effects on the excipient or cause an adverse reaction in that patient. There are two parts in this test, and each part has its own specifications. These are the total aerobic microbial count, or commonly known as TAMC, and total yeast and mold count, or commonly known as TYMC. For the TAMC, a general purpose media is used, and for the total yeast and mold count, a selective media is used. In-process bioburden should not exceed the sterility assurance level of the sterilization process. For example, if you use steam sterilization with a sterility assurance level of 10 to the 6th, the bioburden should not exceed this level. Every formulation must have a validated method, and method suitability tests should be repeated if there's a change in the formulation or the testing method. This is true in any microbiological test method, as we've discussed in previous in 51, or as we referred to in 1227, the validation of microbial recovery. The method used in USP 61s are applicable to in-process bioburden testing for both non-sterile and sterile drug products. The informational chapters, USP 1115, bioburden control of non-sterile end products, and 1229.3, the monitoring of bioburden, provide some additional guidance of the individual products tested details. So through these chapters, they will refer to the other chapters within USP, and we'll be discussing those today too, some of the specifics. There are some exceptions to the sampling requirements. For example, for materials used as active substance where the sample quantity is limited or batch size extremely small, for example, less than 1,000 mils or 1,000 grams, the amount tested shall be 1% of the batch. So in USP 61, the microbial enumeration test, those terms, not less than 10 grams or 10 mils of the product to be tested, are outlined for you. The product is diluted to overcome that antimicrobial property and tested by membrane filtration or plate count method. At that completion of the incubation, those colonies are counted and the results are calculated, and they're calculated based upon the dilution factor for that total aerobic microbial count or the total yeast to mold count. USP 1111 provides some additional guidance on the bioburden limits, and I think frequently questions might come about which organism, and I think these informational chapters provide a wealth of information pertaining to 51, 61, 62, even 71. There are other test methods that can be used for very low bioburden recovery, such as the most probable number or MPN method, and you can find this detailed in the method USP 61. This is another article that Dr. Vu has authored on microbial limits for non-sterile pharmaceuticals, and I believe this is also provided for you in your packet of information for continuing education today. USP 62, the absence of specified microorganisms provides testing method for a list of common microorganisms where absences is required for a certain type of product. As noted in the title, this test is applicable only to non-sterile products. So the absence criteria for many common routes of administration are provided in USP 1111, which is Microbiological Examination of Non-Sterile Products Acceptance Criteria. However, the list is not necessarily exhaustive. Like we've mentioned before, for other microbiological test methods, for a given preparation, it may be necessary to test for other microorganisms based upon the nature of the starting material and the process of manufacturing or compounding preparation. All media used for testing must be verified for growth promotion, growth indicative, and growth inhibition properties for the absence of these tests. The sample size varies depending on the microorganisms being tested for absence. Refer to USP 62 for the specific sample size for each. Also keep in mind that additional samples will also be required when a method suitability test is performed on the preparation along with the test. The test is designed to select for the growth of specific microorganisms. Inoculations, incubation temperatures, and the growth medias used are all geared toward the isolation and recovery of specific or specified microorganisms. When microorganisms are isolated during the absence test, they must be identified to verify the presence or absence of the specified microorganism. This is another article that is composed by Dr. Vu in Quality Control and showing the microbial limits for the specified as noted in Part 2. And remember, if you have any questions as we go along, please submit them during the presentation. I think we'll compile those and then ask the panel of experts at the end. So please, if you think of any question, even related to this, we have a panel of individuals that are there to help to answer that for you. USP 61 and 62. This table 1 is USP 1111 provides that total area, that TAMC and TYMC and absence criteria based upon the route of administration. And again, we said this list is not exhaustive. But the drug product manufacturer should evaluate the necessity of other challenge microorganisms based upon the nature of the individual excipients or the manufacturing process or even the details of the use of the finished product. So essentially, limits as they apply to the test in Chapter 61 and 62 are presented here in Table 1 for you. And I have broken that up into several of the slides. So the next slide is an additional acceptance criteria for the non-sterile dosage forms, and then following up with the third slide. So we broke those up into 3 slides. But this table is this is Table 2 actually from 1111, whereas the previous 2 slides are Table 1 from Chapter 1111. Okay, let's look at interpret the sterility testing results and assess some of the limitations when conducting sterility tests. Sterility test applies to substances, preparations, or articles according to the Pharmacopeia are required to be sterile. This is taken chapter and verse directly from USP 71 in the first paragraph. Pharmacopeia procedures are not by themselves to design to ensure that the batch of product is sterile or has been sterilized. This is accomplished primarily by the validation of the sterilization process or the aseptic procedures. This test, a satisfactory test, results only indicating that no contamination microorganism has been found in the sample examined under the conditions of the test. And I wanted to reiterate that look at which the tests are conducted and understand the purpose. It is critical to validate that sterilization process or the aseptic procedures and processes. Some of the growth promotion confirms the media to be used for the different tests, Candida, Bacillus, Aspergillus. is used utilizing the TSP, and then the fluid thioglycolate, or FTG, is utilized for the media suitable for the Clostridium, Pseudomonas, and Staphylococcus aureus. The method suitability demonstrates the validity of the sterility test by eliminating that antimicrobial activity in the formulation and establishing that the contamination, if at present, it would be detected. So you're trying to minimize or eliminate the antimicrobial activity. Should a bacterium or spore be present, it could be detected. And the idea of method suitability is like a validation or verification. I think they used to call it bacteriostasis fungistasis. But once that method suitability has been demonstrated, it applies that specific formulation and does not need to be repeated unless there is a change in the formulation or the production method or the test procedure. So if there's a change in any of those, the method suitability would need to be repeated for that specific formulation. Next, we have given Table 2, which is indicating the minimum quantities to be used for conducting the test. So if there is less than 1 ml, you'll use the whole container of the contents of that vial to be used in conducting the test, such as 1 to 4 ml in the quantity of the container. You would use half of that, but not less than 1 ml. So Table 2 and Table 3 are listed in USP 71. USP 71 should not be cited as USP 71 if it does not meet these minimum criteria of the amount used in conducting the test as reflected in Table 2, or the number of articles tested as selected and listed in Table 3. And one point that you might know, when capturing, and let's say you're making a large batch, from a practical perspective, you're trying to find, is there an error? Is there a potential contaminant? Is there a problem in the microbiological integrity, breach of contact or integrity? So one way of doing this, and it's suggested maybe doing some of the at the beginning, middle, and end, versus let's say you have a large batch and you just pull some to meet the criteria. Do everything you can to find that error, be conservative in the approach, but sample across the beginning, middle, and end, or top, middle, and bottom. The sterility test, let's look at the interpretation of a sterility test of 71. That sample test media is checked for macroscopic evidence at different intervals during that 14 to 18-day incubation period. If at the conclusion of the incubation no evidence of growth is observed, the product is said to comply with the test. If evidence of growth is found, the product does not comply with the test for sterility. In order to declare a non-sterile test invalid, it must be clearly demonstrated that the failure of the test was unrelated to the product being examined. And we'll talk about that, which leads into, as you'll see, some of the importance of microbial identification, and trending. Possible reasons to declare a test invalid is the data of the microbiological monitoring of the sterility testing facility shows a fault. So the testing facility, if it shows a fault, that's a possible reason to declare the test invalid. A review of the testing procedure used during the test in question reveals a fault. Or worse, the microbial growth is found in your negative controls. After determination of the identity of the microorganisms isolated from the test, the growth of the species may be ascribed unequivocally to faults with respect to the materials and or techniques used in this, conducting the test. If the test is declared invalid, the test may be repeated with the same number of units as the original test on the same lot, same batch. If no evidence of microbial growth is found in the repeat test, the product examined meets the test requirements for sterility. If microbial growth is found in the repeat test, the product examined does not meet the testing for sterility. And I would encourage you to exercise caution in your judgment when evaluating and conducting out-of-specification investigations. This is an article that was published. This is a table, too, that's taken from the review of media selections and incubation conditions for the compendial sterility and microbial limit test. But essentially, it's showing the microbial density. The lower the microbial cell density, the greater the possibility or probability of passing that test. Just as inversely, if you have 10 CFUs per mil of microbial contamination, the probability of passing that test is reduced, or another way to say it is 100% probability of failing that test. This is a good article by Tiffany that is cited by individuals that is talking about sterility tests and its application to pharmaceutical compounding. And that also is an article that's been provided to you. Bacterial endotoxins, USP 85. These test the finished product for the presence of bacterial endotoxins, ensure that level of endotoxins meet that criteria. The bacterial endotoxins are known to cause a pyrogenic reaction in the patient when they're present in a pharmaceutical product at levels that exceed that which has been defined as safe by the FDA. When a product is administered as a parenteral, a patient can give or can receive up to 5 EUs per kilogram of body weight, and as an intrathecal, only up to 0.2 EU per kilogram of body weight. It's important to know, in order to determine the endotoxin limits of a particular drug product and a particular patient, this limit must be calculated into the maximum dosage of the product that will be administered as well as the patient's weight. Thus, the need to have that information to calculate the limits. Endotoxins are byproducts of gram-negative bacteria when the cells die, and therefore they have the potential to be present in sterile and non-sterile drug products that are or have been contaminated with gram-negative bacteria. Per US 85, the endotoxin samples must fall within the pH range specified by the lysate manufacturer, typically 6 and 8. This is done on every sample tested to ensure compliance with USP guidelines. In addition, other potential causes or common causes of interference in the conducting an endotoxin testing is the presence of beta-glucans. Let's look at some areas in the pharmacy operation that require environmental monitoring. Environmental monitoring provides data and confirms the effectiveness of the microbial controls present in that manufacturing or compounding and testing areas. Some examples of controls are presented here, but not limited to: sanitation procedures, your HEPA filtration and airflow, the gowning procedures, and the aseptic technique. USP 1116, microbiological controls and monitoring of aseptic process environments, and USP 1115, the bioburden control of non-sterile drug substances, provide information regarding sampling locations and sampling frequencies in sterile and non-sterile manufacturing facilities, respectively. This next slide is one that is stated in, it's a good diagram that's in USP 797. It gives a good conceptual representation of the arrangement of a facility for the compounded sterile products. That DCA, or direct compounding area, and then you see class 5, which represents that primary engineering controls. And this is the area you're doing your compounding in, most controlled environment, most monitored, and the most stringent areas for environmental control. The lowest allowable level of recovery. And then in class 7 could be your buffer area around the class 5 area, and then class 8, generally maybe an anti-room or an anti-area. But this is a good diagram that can explain conceptually. These tables are table 2 from environmental monitoring, and these are representing the allowable see a few recovery counts in different ISO class environments. As you can see, ISO 5 is more stringent than ISO 7. Table 2 represents, or it relates to air samples. Table 4 refers to surface sampling, such as fingertip samples. These areas are critical where the sterile products are manufactured or compounded. This should include establishing a robust monitoring program, sampling of critical zones and high traffic areas, and even trending of microbial counts and recovery. I wanted to identify if it's something I felt important or was a intentional, purposeful of the chapter, we placed it in blue or italicized it. The isolate should be identified for trending related to the type of microorganism, the level of control in the compounding environment, and the potential source of the microorganism. And remember, non-sterile product manufacturing can monitor the environment with reduced frequency and with expectations of higher recovery. 1113. 1113 is the investigations of microbial identification, or the microbial characterization, identification, and strain typing. Investigations are a major part of any compounding or manufacturing and finished product testing. Any unexpected event, such as in-process test falling out of specification, or an environmental excursion, or a finished product which does not meet the quality specifications that are established, should lead to an internal investigation. There should be a standard operating procedure that describes how to conduct and document the investigation and what information should be collected or reviewed and documented for the investigation. The microorganisms, if detected in a drug substance or water for pharmaceutical use, or the environment, intermediates, the drug products, they typically go characterization and identification. USP 1113, Microbial Characterization, Identification, Strain Typing, provides information regarding the level of identification that is appropriate for microorganisms and that has been isolated during the different phases of the compounding or manufacturing and quality testing. A recent article that was published by Tiffany, as well as Dr. Anstead and James and Lisa, was published in Microbial Identification and Pharmaceutical Compounding. This is a good article, and not only is it discussing microbial identification, but it gives the type and methodology for testing or identifying different microbes. This is one example that might be an output of environmental isolate, where you see the streak of the plate. And then the report might include that phylogenetic tree, or based upon the DNA sequencing, I believe it's the 16S ribosomal, but it's sequencing is to determine that genus species. And so this is an actual output from a report that identifies the isolate that is saying that 99.45 is similar to the Micrococcus luteus. But I wanted you to see this because you can see the isolate may be 1%, so it's important to understand and to be able to interpret that genus species and the data that comes out of that test. Finally, we've given a few but not, references that are important that could serve as a good reference or resource for you. As we just discussed, the many chapters in USP, good articles or journals that are published in various journals such as IJP and other, your testing laboratory. There are many good laboratories out there, but whichever you choose, there are other good experts in the field, such as CGMP validation, Laura Gilligan. There's a microbiology network that's out that Scott Sutton started originally. Andy Gunn, he's another consultant. Compounding Today, the FDA website, as well as the USP website, also provide good resources or references for you. This evaluation and questionnaire link for your CE credit is provided for you on this particular slide. We will now open it up for some of the questions. Okay. here's a question that's going to pose to one of our analysts. The question states, should the chemical manufacturer or wholesaler be measuring and reporting bioburden load on the C of A? So Andrew, Tiffany, or James, should the chemical manufacturer or wholesaler be measuring and reporting bioburden load on that C of A?
That's gonna actually depend more on what their requirements are. So it's probably going to be a case where the supplier may or may not supply that information to you. But as a good practice for your own business, you're going to definitely want to perform that testing on your own, whether they do or don't, to confirm the data.
Thank you, James. Another question is when would you compound a non-sterile inhalation? Andrew, Tiffany, or James, when would you compound a non-sterile inhalation?
Well, when it's not a solution, then for example, dry powder. Even dry powder, sometimes you have to do it in the sterile format. Whenever it's in solution, then the FDA recommends that it should be sterile prep because the past incidents of contamination of the nasal prep that lead to what you call meningitis, and so it's important to keep the solution sterile. Or if you do multiple dose, then it should have antimicrobial preservative in it, and you need to demonstrate 61, 62 acceptable at time of release and also at the end of the beyond use day.
Thank you, Dr. Vu. That was excellent. Another question is, what should happen if a product fails method suitability, meaning that all the specified organisms would not grow? What would happen, or what should happen, if a product fails the method suitability, meaning that one or all of the specified organisms would not grow?
This is James again. USP 71 gives some really good guidance on that. It would basically mean a modification of the test method, and that can be the addition of some kind of neutralizer for anything that is suspected to be preventing growth of those organisms, an increase in dilution if it's a direct inoculation method, a change of filter if it's a filtration method, or a difference in volume or type of rinse fluid. So there's a number of different measures that the lab can take to get a sample to pass those, to come up with a reliable method for establishing sterility.
Okay. Thank you, James. Does USP 795 state that we need to do sterility testing on non-sterile products? Does USP 795 state we need to do sterility testing on non-sterile products?
No, the chapter is designed just to cover those products that are expected to be not sterile based on the factors that were presented in the slideshow, and then they would just fall under the testing required under that chapter.
Okay, thank you, James. How often, there's a lot of questions here on me. How often must one perform 61 or 62 tests, and must we perform these microbial enumerations on every compound? How often must one perform USP 61 and 62 tests?
It depends on the individual compounder's SOPs. It's always, I mean, the more data you can have on the quality of your processing, the better. But it's going to depend on how often the requirements are set by the place where the compounder is located, and their own quality control SOPs.
Okay. Thank you. Let me see. Get back to here. There's a lot of questions here. What is the minimum volume considered large volume parenteral? What is the minimum volume considered large volume parenteral?
The way 71 is interpreted is generally 100 mils is the cutoff. So up to 100 mils, and then anything above that is a large volume. So commonly that's going to jump up to those 150 or 250 mil bags and above, but it's usually about 100 mils is going to be the cutoff.
This is a good question. Is random sampling of sterile products appropriate, or should we take a sample vial from the start of the compounding, middle of the filling, and end of the filling? That's I think pertains to process verification, process validation. But the question states, is random sampling of sterile products appropriate, or should we take a sample vial from the start of compounding, middle of filling, and end of the filling?
Hi, this is Tiffany. For sterility testing, you always want to test your finished product, so it would not be appropriate to take fills from the beginning, middle, and end of your process. You would want to complete all of your fills and take random sampling of the finished product that has been filled into its final article.
That's a very good, important point. 71 is intended for finished product testing, for the finished product. And that's not to ignore the fact that you should verify and validate your processes, which is what the first thing we stated and put in bold and in red. Let me go back here. Okay, how would you count a large colony on a surface plate? How would you count a large colony on a surface plate?
Regardless of the size of the colony, because it's still going to be counted as a single colony. If it starts with multiple, you won't be able to tell by the time you're reading it, so you can't try and interpret or guess anything. And that's more about the growth rate of the bacteria than having multiple colonies. So you're still going to count it as one, no matter the size.
Okay, excellent. What if, during environmental air monitoring, the cumulative amount of the microbes present on several plates exceed 10 to the question mark? Does 10 apply to a single plate only? I don't quite understand that question. What if, during environmental air monitoring, the cumulative amount of microbes present on several plates exceed 10 question mark? Does 10 apply a single plate only? I think that's what they're saying.
It's, of course, individual SOPs will vary, but it's usually per sampling event, so per plate. Now that may be something where if you put out multiple, say, passive plates, you add up their total, but it's going to depend on the SOP of the pharmacy. So in some cases it may be a single plate, in some cases you could add them together. Something like that.
Okay. Another question, thank you, James. Is the color of the colony significant?
No. There are objectionable or pathogenic organisms that have plain white colonies, in the same way there's completely benign organisms that have plain white colonies. And red, they come in all colors, so red, green, yellow. Any of them can be pathogenic or not. And that's why it's a good idea to have them identified, so you can be sure, because visual identification only takes you so far. Identification to the genus or species level is really what you need in that case.
What instrument do you use for the endotoxin testing? What instrument do you use for endotoxin testing?
We use a kinetic turbidimetric reader. There are gel clot and other methods out there. It's usually some kind of plate reading device that you can run multiple through at the same time. Although there are kits out there that let you do individual samples, just depending on the need of the testing lab or the pharmacy. So you can run one sample at a time, or you can run 20 samples at a time. It's definitely a scalable type of test.
Thank you. When you have the genus and species, where do you look to see what may be the source of this microbe? When you have the genus and species, where do you look to see what may be the source of this microbe?
Resources for where the microbes might have come from. Berge's Microbiological Manual is a good one. It's kind of the Bible for that type of organism information. But online is a good resource. There's a lot of good websites out there that have a lot of information as to possible avenues of entrance and where the organisms you're finding in your facility come from. So just you can get quite a lot of information from a little bit of research.
Okay. Is there an environmental monitoring requirement for non-sterile areas? Is there an environmental monitoring requirement for non-sterile areas?
So those ISO 7 and ISO 8 areas, it's going to more be dependent on the ISO class of the compounding area than whether you're doing non-steriles or steriles. In any case, you want the lowest possible bioburden entering your product. So it's a good idea you don't want to recover anything, but if you do, it's more about what your compounding area is classed at as to whether it meets or fails any kind of criteria.
For non-sterile dosage forms, what type of microbes should be tested? For non-sterile dosage forms, what type of microbes should be tested?
As far as what genre or species you would look for, USP 62 organisms, those are all going to be good representative organisms, their various groups. Guidance for that is in USP 1111. But it can be, for example, your environmental monitoring can tie into what organisms you test for in your non-sterile compounding. If you have an organism that you often find in your environment that you would then like test it for to see if it's the frequency with which it enters your product, you can screen for that organism in addition to what is listed in 62. But 62 is a good place to start. And you're just going to have to evaluate what organisms are an objectionable thing to have in your drug product based on, its end user and its application.
There's a follow-up question, and it says, is 61 or 62 testing mandatory per USP regs? Is 61 and 62, USP 61 and 62 testing mandatory per the USP regs?
They're both sub 1,000 chapters, so they're enforceable. That's the short answer for that. Got to make sure and meet those requirements.
Excellent. How frequent do you perform sterility testing, with every batch or randomly? How frequent do you perform the sterility testing, with every batch or randomly?
Every batch would be good, but it depends on your aseptic processing validations and your internal SOPs.
Is there a comprehensive list of pathogenic bacteria?
Not comprehensive, but the FDA actually puts out it's funnily enough called the Bad Bug Book, and the FDA actually publishes that, and it's their list of common objectionable organisms. So that's a really good resource. It's a good read. A lot of information about kind of tying back into that other question about which organisms to look for. The Bad Bug Book published by the FDA gives a pretty good description of a lot of organisms that can be pathogenic.
Thank you. In the case of non-sterile compounding, when is it appropriate to conduct USP 51? In the case of non-sterile compounding, when is it appropriate to conduct USP 51?
During your formulation development, and the guidance is good for anything that's a multi-dose a drug with a preservative in it. You want to be sure and verify that the preservatives are going to do their job, and that's exactly what the USP 51 test is designed to do. Ensure efficacy of the preservative throughout the time you're wanting to store it or the patient might use it.
Right. That reminds me of that. Dr. Vu wrote an excellent article on that USP 51 that was included, I think, one of the first ones that we discussed. Is there an ISO requirement for non-sterile compounding? ISO 7 or 8 are typically your buffer and antirooms in your sterile suite. This is a question. 1, is there an ISO requirement for non-sterile compounding? And 2, ISO 7 or 8 are typically your buffer and ante rooms and your sterile suites? Questions.
You want to compound your non-steriles in as clean as environment as you could. So ISO 5 would be preferred. I would say that's a good chance for you to explore your results from your 61 testing, to establish levels of process control that you have in place, and then determine whether what you've got set up is meeting the requirements.
Okay, it's approaching the 1:30, and that concludes the last question that's been given to us. We really do appreciate everyone's participation and the great questions and the team of experts that were there for the answering. Amy, do you have any following comments before we conclude this webinar?
Thank you all for your time today. Following the webinar, within 48 hours, you'll receive a recording link to this session, and it'll also include question and answers at the end. Following up to that, we're going to compile all of the questions that were asked today along with the answers, and we'll include a FAQ document. That might be first part next week. Remember that in order to receive your continuing education credit, you do have to complete the questionnaire and evaluation link, and that is located on the handouts. We'll also include that link in the follow-up email to the attendees as well. And thank you all very much for your time.